overall goal of this procedure is to use electroporation to introduce siRNAs into difficult-to-transfect cells in order to study gene expression. This is accomplished by first determining the amount of Namalwa cell suspension needed using the TC10 automated cell counter. siRNA is then added to the cell suspension, and cells are electroporated using the MXL electroporation system. Cells are incubated for 24 hours post-electroporation, then IL-4 is added to induce gene transcription in experimental cells. Finally, RNA is extracted, and real-time PCR is used to measure levels of gene expression. Ultimately, results can be obtained that show the effect of specific siRNAs on gene expression pathways.
Hi, I'm Adam McCoy, Senior Scientist at the Gene Expression Division of Bio-Rad Laboratories. Today, we'll show you a procedure for introducing siRNAs into difficult-to-transfect cell lines by electroporation, and then look at the effects on gene expression. We use this procedure in our laboratory to analyze gene expression of single genes or study gene pathways by knocking out one gene and looking for downstream effects. So let's get started.
Working under a cell culture hood, remove the cells from a culture flask. The cells used in this protocol are suspension cells. If you are using adherent cells, you will need to trypsinize before removing them. Centrifuge the cells to pellet them. After the cells have been spun down, remove the supernatant and resuspend the cells in PBS. To determine the number of live cells, take an aliquot of the cell suspension and add trypan blue stain at a ratio of 1:1 to stain the dead cells.